Engineering from, right, from the Rochester Institute of Technology. So he's currently on sabbatical in San Francisco, and he's going to tell you a little bit more about that, or you can ask him during the reception. He does some really cool work at the intersection of AI, biochemistry, and drug discovery. And I heard him talk at one of the NIH IDG meetings, and I felt, okay, we need to get him here for the symposium. So he's published over 50 papers in this area. He's one of the few people who really works at the intersection of AI, biochemistry, and drug discovery, and you'll see some of the cool stuff that he does. But more importantly, he was one of the few people, who's sort of the red teamer for OpenAI's GPT-4, model, and hopefully he'll tell you a little bit more about that. But he also serves on the advisory board of multiple AI companies, and I think today he's going to tell you about some of the work he's doing, and the title of his talk is From Intent to Compound Chemistry with Agents. So welcome, Andrew. Yeah.
All right, thank you, everybody. It's been a really exciting time the last, say, 12 months or so. I read this like metaphor about what it's like right now in AI, and somebody drew an analogy to like first contact with aliens. Is like, we train this model on all of human knowledge, and then we took it out of the model training environment, and then we asked it some questions, right? And we've been like learning the language of this alien model and seeing what it's capable of doing, right? Like there was a very famous researcher who had worked on, I think it was meta learning and reinforcement learning algorithms, very complicated topic, very intense math. And then his like latest claim to fame was the idea of let's think step by step, just like putting that sentence in front of all other instructions to these LLMs made their accuracy shoot up by 15 points. And so it's been a remarkable time. And I'm going to share with you some sort of, I would say, very early work. Everything being done right now is very early work with these LLMs, these foundational models. But I feel like it's almost criminal to give an academic talk only about that, because there's no principles, right? A lot of it is like, we tried this, it worked pretty well, and I can't tell you why or how. So for the first part of my talk, I'm going to try to give you all some principles of deep learning, for you students in the audience, some principles of deep learning, and then I'll show you how they all break immediately. All right. So I first want to introduce my lab group. We are way overdue for a picture, but this is back during the pandemic. I'm slowly, when I was a young professor, I was like, I'm going to update my website all the time. There's always going to be a fresh picture of us, and I'm slowly, ossifying until this will probably be the final picture of me for the next 50 years. But all right. So I want to first say I wrote a textbook called Deep Learning for Molecules and Materials. This is an introduction to deep learning for those of you who are like a biologist or a chemist, somebody coming from a natural sciences background wanting to learn deep learning. So this is written for people also who want to know, graph neural networks first. If you learn deep learning in a computer science course, graph neural networks is maybe chapter 13, some obscure topic, and then image and audio, and speech are the #1 topics. This is sort of graphs first, because most things in chemistry, and I think biology too, although of course there's these sequences everywhere, a lot of things are graphs. All right, so like I said, I want to talk a little bit about the principles of deep learning. So the first concept I want to introduce is something called an inductive bias. And this is kind of like a catch-all word for decisions we make when we build a neural network. Okay, I don't even have a slide on this, but remind ourselves, in machine learning, we have data, which is made up of features and labels, X and Y. And the goal is to find a function that generates Y from X. Okay, sorry, I forgot the slide, but I think hopefully you all know what machine learning is. Now, deep learning is when we make sure that that F of X, that function we're trying to learn, is a neural network. And then an inductive bias is any extra information we know about F of X. How do we apply it? Okay, you can also think about this as like, decisions or things we know about the physics of the problem. So I'm going to talk about chemistry because I am more of a chemist than a biologist. And the first kind of inductive biases come from how we choose to represent data. So if we represent a molecule as a graph, that says something about the order of the atoms. That is, the order and how I write down the atoms doesn't matter, because in a graph, the order of the nodes in a graph are relevant. But if I write things down in a point cloud, right, then maybe the XYZ coordinates matter, but on the left-hand side, there is no XYZ. So if I rotate the molecule, it doesn't mean anything on the graph. And I've recently learned that DNA also has a similar symmetry property, is that DNA can be shifted by 6 ways. You have your reverse complement, your frame shift. So if you're ever doing reads, metagenomics, for example, you have to think about the different ways that DNA might be basically operated on by a symmetry operator. Okay, now let's go into some of these details. So what this actually means is we have these things called equivariances. Equivariances are the same thing as symmetries. So like, like left-right symmetry, maybe from certain molecules. There's also something called atom permutation equivariance. It says that if I swap the order of atoms, like if I write down the nitrogen first in a list versus the carbon, it shouldn't matter. So I have a little example here. So let's say I have a neural network. So remember, I'm basically inputting my graph as X. And what I'm doing here is I'm showing you, I'm representing my atoms using something called a one-hot vector. This is a very common approach in machine learning where I have my carbon atom here being indicated as a vector 1, 0, 0, whereas my oxygen is the vector 0, 1, 0. And I'm predicting something like partial charge or electronegativity, something like that. Now, if I swap the input to my function, like I swap the order that I wrote down the atoms, my output should also swap. Really simple idea. It turns out the only kind of function which can do that is a graph neural network, okay? This is kind of a trivial thing, you can also sort of call deep sets. The only thing that can do is a deep set, but the definition of a deep set is that it does that. So it's kind of a ridiculous proof. But if you want to get this neural network, you can't use a convolutional, you can use graph convolution. You can't use an image neural network. You can't use, a standard multi-layer perceptron. Same thing with geometry. So if we have translation or rotation, as my neural network operates on these things, I need to make sure the output, here the output is the red lines here. As my molecule translates across, I need to make sure my output translates with it. So this is another example of an equivariance. Okay, now these equivariances are tied up to the data we have, and these equivariances are tied up to the model that we choose. So if we use a transformer, for example, that has an equivariance in position. And in fact, one of the strangest things about transformers is that the great thing about transformers is that they're equivariant with respect to position. But then the first thing people do in modern transformers is they break that equivariance by putting a position encoding back in. It's kind of interesting thing about transformers. Whereas if you use a recurrent neural network, then position can be put in, cannot be put in. Graph neural networks are how we do the atom permutation and bond permutation equivariances. And then equivariant neural networks is kind of a catch-all term for these higher order geometries that we can capture. Okay. So there's also a connection between the properties. So if we want to predict a molecular property or a specific atom property or a specific point in space, then that also has an impact on what equivariances we choose and therefore what neural networks we choose. Okay, now these are all very exciting and there's lots of fun math, but I want to talk a little bit about just text, right? So one of the cool things about text is that it has reached a maturity level where now you can run very big multi-million parameter models in your phone or on your laptop without any effort at all. So this is a neural network where you put in a peptide and the neural network is running on my web browser and will predict properties. So we have actually 3 neural networks that then predict if this peptide will be hemolytic, if it will be soluble, if it will be specific or non-specific, basically will it aggregate and be immunogenic, and how easy would it be to synthesize. So this is a bunch of neural networks that can run in the laptop, and you can sit there and type and adjust your peptide sequence. So it's really remarkable that we can do a lot of stuff that was very difficult, required a supercomputer. You can now do it in your browser. And I think we're getting to this point now where there's a lot of areas in biology and chemistry where the amount of data and the size of the models doesn't need to be large. Like we don't need a foundational model for every task. So I think we might be seeing more of these sort of in-browser, very simple models that are easy to use and don't require a lot of effort. Okay, now let me talk to you about the state of deep learning in molecules. The graph neural network. If you go to any pharmaceutical company that's doing drug discovery, this is what they use. The pre-trained graph neural network is the state-of-the-art, the ubiquitous piece of deep learning. I'm not going to show you a lot about graph neural network. I made a very nice animation of how they work. Let's just take a moment to appreciate it. Very cool. And then we'll move on. Okay. And I'll tell you why we're not going to be learning about how graph neural networks work later. Now, here's one interesting thing is that these are 2 molecules, decaline and bicyclopentyl. These are 2 molecules that are different different synthesis, different properties. But if you actually look at the nodes, like if you look at the nodes themselves, it's a red connected to two greens here and a red. This is a red connected to two greens and a red. You'll notice that they actually look the same to a graph neural network. And so it turns out that molecules are not exactly graphs in some ways. Now you can fix this using higher order graph neural networks or using simplicial complexes, doing fancy math. But then you have these molecules like this, which has global stereochemistry. That is, this molecule can either have a clockwise or a counterclockwise turn, but that is not at some specific carbon. It's not, there's no center of stereochemistry in there. It's a whole molecular property. So this just breaks any kind of graph neural network that you can come up with for representing this correctly. So what do? So what happens is that people then start including geometric information. So we have to do things like build equivariant neural networks that include the local 3D environment. So we learned about this in the spatial transcriptomics. And then we also have to think about things like passing information in the graph that includes geometry. Now, what is the, this is the body order. So basically you look at your local neighbors. Now, the way that we encode geometry is something called spherical harmonics. They are the irreducible representations of three-dimensional shapes. Okay, so the way that we send messages in graph neural networks, not as just the distance between things, now it's the orientation and the higher order spherical harmonics. And you see these neural networks that are called equivariant message passing neural networks. One of the fun things about spherical harmonics is that as you increase the order, the dimension changes, right? So we have these very unusual tensors that are called, oh God, what are they called? I'm blinking. Fragments. We have these things that are called fragments where you have like a 2 and a 3 and a 5 and a 7 is the dimension of them as you increase. Very hard for computers, then they're very slow. People are working on it, it's getting better. But then you look at the field, now there's all these different ideas about how to encode this information. And I have bad news for everyone, okay? So we can solve this. As a community, we're almost there that we can encode three-dimensional information, but then people are gonna find out that we have dihedral angles. And dihedral angles on molecules means that they're not always in the same shape, right? And then people are going to find out about these things called alpha helices and beta sheets and realize, that you can't just represent it here. And then they're going to find out about, electron density. And it turns out that molecules are not always the same. And then the worst part is, did you guys know there's no definition of a chemical bond? There is no quantum mechanical observable of a chemical bond. Nobody even knows what a chemical bond. It's just some hallucination that we all agree is a good mental model, right? But there's no measurement. You can't go out there and just, oh, is this a chemical bond or is this a chemical bond? If you think you can, look up the molecule ferrocene, break your brain. Okay, but does it all matter, right? So here's an interesting example, okay? In the AlphaFold paper, they put an invariant point attention module. So this is an equivariant attention that they added to it. And this is a lot of work, it's a lot of math, but if you take that out, what happens to the accuracy of the AlphaFold model? Well, not a whole lot. It goes down by about 1 or 2 points here, depending on which point you're looking at. So this is the ablation. Turns out the equivariance is a lot of work and it doesn't really pay off that much. Now, with that said, equivariance is required for many things. Like, if you want to be doing diffusion models for protein folding, you have to have equivariant models. But it really is, I think, overemphasized. So I have this hypothesis and I'm going to make a better picture. This is a placeholder, okay? So yeah, next talk, I promise it'll be better. Okay, here's the idea. When we actually make something, okay, there's a few things we can control. We can control how we make it. We can control the ingredients we order and the processing. Then we have this structure, and we can disagree whether, we should be thinking about the DNA, the RNA, the amino acid, the primary structure, the tertiary structure, the quaternary structure, the rotamer library, whatever. But at the end of the day, that's just some mental model that we all have to grab onto. And then we have the measured properties in the lab. Now, I want to show you that we can actually drop that middle part, that we don't actually need to have that structure as we don't need to put such an inductive bias on what that looks like. So I'll just give you like some early hints of this work. So there's a paper from December, and this one showed that if you take tabular data, in this case, a very famous tabular data set, where we try to recognize what's the species of some flowers based on properties of the flowers. If you take the data out of a table and you put it into an English language sentence, and then you fine tune a language model on the English language sentence, you can do classification, okay? So we can take data that's in a table and put it into an English language sentence and get good classification accuracy. And then Kevin Jablonka, Baron Smith, Philippe Schwaller did the same thing in chemistry now. Now we're saying the solubility of methanol is blank. Okay, now we're taking English language sentences about solubility, fine-tuning a language model and getting good performance. And in fact, if you look around a whole bunch of tasks, doing this language fine-tuning, this like GPT-3 here is the model, it does better than most other bespoke deep learning models at fewer data points. So if you have 50 data points and you don't know what to choose, you can take a language model, fine-tune it on your task, and it will often be doing better than any other models out there. Okay, I'm going to just kind of go through this a little bit quickly because I think it's not that exciting, but basically you can go farther and you can get this to have uncertainty. How do you get it to have uncertainty? There's a few ways to do it. One kind of interesting way we figured out how to do it is that if you ask something like the solubility of methanol is blank, and then you turn it to multiple choice questions, you know, A, B, C, D, E, and then you have the model predict what's the answer, that gives you a discrete probability distribution because language models give you a per token probability. So it'll give you the probability of A being correct, B being correct, C being correct, et cetera. So you actually get uncertainty now out of these language models. So here is some results here. I'm showing you the solubility of 1-propane is minus 1.73 log S. So we've taken English language sentences, and then we basically are predicting the solubility with these. And you'll notice that I have error bars here, and that's from this way to get uncertainty out of it. And I'm showing you a few different ways to do this. So DaVinci and GPT-4 are both language models, and here we're using in-context learning. That is, we're just giving it preview examples of solubility and asking it, you know, what's the next solubility, and it's predicting very good. These correlation coefficients, 0.92, these are the best, you know, you can really get in solubility prediction. Then fine-tuning, this is where we fine-tune a language model from scratch. So we fine-tune a language model on these data. And then Gaussian process regression on embeddings is just a baseline that I think is very interesting, but we don't have time to talk about it. Now let's go to the, you know, all the way there. Now I'm taking a English language sentence of my methods. This is like the methods section from a paper, okay? I'm putting in the ingredients I ordered, the steps I did, the processing I did, and the reaction conditions, and then I can predict the outcome of the experiment here. What's really powerful about this method is that this allows me to have things like the ingredients and the processing and the reaction conditions all in the same representation, and I can predict the outcome with uncertainty. So this is a really powerful method if you are in a domain where you can't perfectly represent your data as a sequence or as a graph structure or as some kind of, you know, rigid object. There's a lot of exciting stuff in this work. I just want to highlight one question here is that, does it learn, right? So that's one of these things about GPT-4. You go to GPT-4, you try your problem in GPT-4 and it doesn't know the answer. Well, that's it, right? Like there's not really a lot you can do. Maybe give it a few examples. But using in-context learning combined with a trick, what we do is we pull up the most relevant examples of previous experiments we've seen and put them in this in-context. We can do learning out to like 500, 700 examples, okay? Without retraining the language model. So this looks like very interesting code. So what we do is we have code where we say we tell what the X is as a string, and then Y, what the value is a number. And then we can predict on new values. And then I didn't really talk about it because I don't have time, but we did Bayesian optimization. The whole point of getting uncertainty is to do Bayesian optimization. If any of you are in the kind of business of designing things, Bayesian optimization is the best way to design experiments, the best way to design some, you know, new material or something. You can do Bayesian optimization with this method. It's really remarkable. Okay. I have like one example. So this is catalysis is what we're doing here. So we actually did this method. So we used it to make new catalysts, went in the lab and tested them. We were able to get within 2 points of the thermodynamic maximum within just a few experiments. So this really works on catalysts. And I think there's, you know, I don't know, I'm still learning about all the domains of biology, but I bet there's other places in biology where you can represent your experiments with language. Okay. So this is crazy, right? Like, you know, what does this mean here? So why would large language models be any good at this? Well, part of the reason why is that GPT-4 is not like a, you know, it's not like a breakthrough in all of physical sciences and mathematics and everything. What GPT-4 is doing is basically absorbing everything that we've taken the time to write in language. Basically, every time we've had a new physical phenomena, every time we've invented a new experiment or invented a new field or saw something that is unexplainable, we created language to explain that phenomena. We made language match our physical reality. And so it's kind of like a two-step process. We all collaboratively created language over thousands of years to represent the world, our culture, ideas, what we think of as science. And then GPT-4 learned to absorb that. And that's why we get all these emergent capabilities out of it. So it's not really that LLMs are somehow special, it's that language is somehow special. Okay, so this is my hypothesis that language is the future of chemistry. I wrote a little article about it. You can read it, it's very short. You don't have to read it because I'll talk more about it. Okay, so the first thing I want to talk about now is that I think we're starting to get a handle on using language. We can represent experiments of language. We can basically capture new kinds of regression properties of language. But what is the biggest source of language? I believe it's scientific papers. Basically, that has been somehow, I don't know if we all agree on this, but we all follow this idea that the deliverable, the outcome of science is a paper. And so if you think about it, a paper is the real, I don't want to say the raw data, but it's the refined data from thousands of years of science. Sorry, I don't know, hundreds of years of science is in papers, right? So I want to talk about our first project, which was how do we tap that information inside of research papers with LLMs? Okay, so let's start with some question answer. Okay, so here's a question that I've asked GPT-4. Which three residues with evolutionary divergence in the G domains of RAS isoforms also impede selectivity constraints on pan-KRAS non-covalent inhibition? This is a cancer drug discovery question. Anybody in the audience know the answer to this one? Yes, well, don't worry. GPT-4 gets it completely wrong with hallucinated answers, hallucinated references. It's all garbage, right? Darn, okay, so we tried the easiest thing, it didn't quite work out. Okay, so it turns out if you go to these language models, like GPT-4, GPT-3.5, Claude 2, they hallucinate. How often do they hallucinate? Somewhere between 30 to 50% of the time. So you ask them scientific questions, we get hallucinations pretty often. And so there's this problem, it's got a name, it's called the bullshit asymmetry principle, is that it's easier to generate bullshit than it is to disprove bullshit. And so although GPT-4 might, you think, is helpful to you, it actually generates a whole bunch of crap that you then have to figure out if it's real or not, right? So it's actually takes longer to verify the responses than it does to generate them. Okay, so here's how we can solve this, right? So again, I believe that language is how we can solve all problems in the world, right? Okay, so but how do we solve this specific problem, right? So what we do is that rather than relying on GPT-4 of having read all scientific papers, when it comes time to answer a scientific question, we let it cheat. We pull up all the scientific papers we think could be relevant for it, and we give it as extra information. So I ask it this question, this difficult question, but then I pull up all the papers that could be related to this question, right? And so now we say, which three residues are there? And then we get back the three residues with evolutionary divergence in the G domain of RAS isoforms that impose life constraints. They're 121, 122, and 95. And now the paper it cites is a real paper. Why is it a real paper? Because I cheated. I gave it all the papers I thought could be relevant, and it chose out of those papers which one was the right one. And so if you follow this process, you get zero hallucinations because everything that it answers, every reference it generates, I tell it, do not guess, you don't know, only respond using the information I've given you. Okay. So that's great. So we can now kind of tap scientific literature. We're going to go back to that project in a minute because one big question is like, how do you get the papers? Of course, it's the same question I've had all my life, right? It's like, which papers do I need to read? But let me talk to you now about going one level deeper. So one of the benefits of language is that the way that we think about using tools in science is with language. If you want to use a new, say, bioinformatics package, you don't read the source. I mean, I'm sure there's one person in here who reads the source. Good for you. But most people read, the documentation or the paper, or they ask their friend, what is this package for, right? So a lot of times the definition of a tool comes from the language that describes it, the natural language. And there's been this idea that if we take natural language and it can be our glue, then we can start bringing in tools like a calculator, a bioinformatics package, a Python interpreter, and we can have LLMs use these tools using language as the bridge between them. And this is called agents, okay? Let me show you a little bit about how this works, okay? So I want to talk to you about chemistry with agents. So what I'm going to do is I'm going to have an LLM and I'm going to tell it, you have the ability to use tools if you're not sure. So when are these things not sure? I I know that GPT-4 can't count the number of like hydrogen bond donors in a molecule. It just can't do it. So I tell it, you don't know chemistry, use the chemistry tools. Don't try to work with chemical structures directly. So then I give it some tools, web search, a PubChem search, an LLM code model. That is, my agent can give instructions to another LLM, which then writes code that then executes the code and returns a result. A calculator, synthesis planner, and paper QA. This is this thing what I told you about, is that it can go read papers, get back information from the papers to then go forward. So I give it a question, design a compound with the same mechanism as dasatinib and purchase it. Okay, so I'm not going to read all of it here, but I'll just cover the highlights here. So I've asked it to propose a compound with similar properties to the drug dasatinib. So what happens is that I tell it to use this pattern. I tell it to think in a question, thought, action, action input, and then reflection / observation. This pattern is called REACT. And so what happens is I can see how the LLM thinks through it. So this is all written by the LLM. So it says I need to find similar compounds. It uses the literature answer. This is this paper QA tool. Then it asks the tool, what are some compounds with the same MOA? And then what happens is it brings back some compounds with the same mechanism of action and the same target as dasatinib. Okay. Now it calls up a different tool. It needs to get the SMILE string. Remember, I told it that it doesn't know chemical structures. So now it's going to turn those names of compounds into chemical structures using a tool. So it comes up with imatinib. Then I tell it to modify the compound. Now, what this modification is doing is it's escaping a patent. Okay, so this compound is patented. I'm telling it to escape a patent. Okay, this is not how you do drug discovery, okay? This is just a concept here. Please don't go out and do this. Okay, so then we come up with a compound which has been modified so it's no longer patented. Then it checks to make sure that the compound is purchasable. Then it finds that we have a tool which goes to look in chemical suppliers, and it finds that this compound is purchasable, so then it purchases the compound. So at the end, it says, I know the final answer, the compound blank, it's like an imatinib slight isoform or something. I forget the word is. It's like a stereoisomer of imatinib in a weird way. It's a novel compound with properties similar to dasatinib that's been purchased from zinc. We did find out later on that it was actually patented by a Chinese pharmaceutical company. Our patent database didn't include Chinese patents, but it was pretty good first guess. Okay, so what I've shown you is that we can take LLMs, we can give them tools. And remember, they know language, they know natural language. So as long as we formulate our tools to work with natural language, then we can have them use these tools. So then we wanted to see how far could we go. We went as far as we could and brought out our collaborators at IBM. We got a robotic synthesis platform that could synthesize novel compounds. And then we expanded our literature tool. And then we're able to do, I'll read you some of the things that we're able to do. Make an insect repellent. Okay. So then it went and found the structure of DEET on Wikipedia. It then looked at the available compounds that the robot had. Then it proposed a retrosynthesis plan for the robot. It executed the retrosynthesis plan on the robot. Then it read the FTIR spectra, a mass spec that came out of the other end of the reaction, and it created a small amount of DEET. Okay. Make a thiourea organocatalyst. That's a bad name. And then it went to find a catalyst for this reaction. It planned the synthesis or automated it. So I think the most impressive one, though, is the last one, is where we gave it the supporting information from a paper, and that supporting information showed some compounds and the colors of the compounds. So it's basically like making a dye. And we told it make a new dye, and we gave it the exact wavelength of color we wanted, okay? So then what it did, oh, I don't have this example, darn it. It's a problem, okay, I'll just walk you through it. So then what it did is it took the supporting information from the paper, it loaded it up into Python, then it trained a classifier with sklearn, with scikit-learn. Then it used a classifier to predict a bunch of compounds it could make until it found one that was close. Then it went to go make that compound, and it was a two-step reaction, and the robot kept failing. I hate robots, okay? And the organic chemist watched the robot fail like 3 times in the week, and it was just ridiculous things like the needle wasn't lined up, and the organic chemist was fed up, and so they took the recipe and then synthesized it in the hood. So then we had to change that section of the paper to be AI human collaboration instead of AI robot collaboration. But then we made the molecule and we tested it. It was within 25 nanometers of the desired wavelength. So I think we wanted like 545 nanometers. It was like 565 or 570 nanometers. So we were able to go from take this data, make me a new compound with this specific wavelength. And it built the machine learning model, it executed the synthesis mostly, tested it. It's really remarkable. Okay, and I'll give you one more example. How much time do I have? I have 10 more minutes. Okay, all right, great, no problem. I'm ready. All right, here we go. So here's another example of one of these things. So synthesize a similar molecule to paracetamol that doesn't contain a methylamide group, then find the price of this molecule is less than $500, plan a synthetic route. Another sort of example of how this model thinks through it. So it follows these steps using these tools, going through this thought, action, input, observation. Now, at the end of all of this, we need to like assess how well it did. And this is the hardest problem right now, is that it's really difficult to just say make an insect repellent and then us go and check that it made an insect repellent. That's not really a repeatable evaluation. Like, of course, it's great, and that should be the definition of success here, but it's a really challenging thing. So what we did instead is that we basically had expert chemists evaluate the plan and evaluate how thought, basically give it a number of how well they thought it did. These results are a little bit hard to read, but basically, when you give GPT-4 tools, it does a much better job. If you tell GPT-4 to just do it without tools, it hallucinates all kinds of stuff and the plan is not good. Okay. All right. So I want to just talk briefly about agents of agents, right? So I just described to you that this agent can now orchestrate different chemistry tools, use robotics to make compounds de novo. But one of the problems is like, how do we know which papers to use, right? So what we do is we now have that agent, which is orchestrating chemistry, can call another agent, which can do scientific research. So this agent can search for papers on things like Semantic Scholar or Google Scholar. It can then gather evidence. The way it gathers evidence is that it reads through the papers one page at a time using an LLM and identifies parts of the paper that could be helpful to answer a question. And then it organizes those evidence, those fragments, into a database. And then what it can do is answer the question. Then it calls on another language model, and that language model has said, we have gathered these pieces of evidence. Please answer this question using the evidence. And then it can decide, did we answer the question? No, go back and get more papers. No, go try a different way of gathering evidence. And what this can do is basically answer arbitrary scientific questions. Here's an example. What is the molecular origin of the glass transition temperature? And sorry, the glass transition in polymers. Provide the predominant hypothesis and an alternative with citations, and it will provide to you, answer your question and give you citations, like I said. So what we did is we did head-to-head. So we gave humans 3 hours access to Google Scholar. They can call their friends. They can use the internet as much as they want. And they were given a test to take. And then we gave it to our model to take. The humans got 67% accurate. We got 69.5% accurate. Humans and us both had about an 88% precision. Okay. This test we made was a very difficult test. It is all recent papers. It's information that is contained inside the papers and from multiple papers. So we try to get something that is really difficult that would require careful research. And our model, you don't have to give it the papers. It starts with literally nothing. So it goes out, reads the literature, finds the papers, downloads them, reads them, and answers the questions. And then we have, just for reference, a few other commercial products that claim to do this and some other agent models out there. And basically, we've matched human level performance, but it's now scalable, right? We can run 1,000 of these things concurrently. We can run as many as we want cheaply. We don't have to wait 3 hours. So we basically automated at the level of humans this ability to do scientific question answering. And who were the humans? They were all MD, PhD students from Oxford. So I don't know what this is about Oxford, but there you go. So I also have some other results. So people always like these sort of these benchmarks, and I'll just share these. So one of them is like PubMed QA. So PubMed QA is a pretty common benchmark for question answering of models, but it's too easy. So what we did is, usually in PubMed QA, it gives you a question, it gives you context, you have to answer it. We deleted the context. So it just gives you a question, you have to find the paper that you would need to find to answer it. So like GPT-4 gets a 58%. We get an 86% on this. And then of course there's the MedQA. Now interestingly, we don't see a big gain in MedQA, USMLE. And that's because like most of the information for diagnosing patients actually comes from medical textbooks, not necessarily from research papers. So this is kind of a nice control. Like we're only looking at scientific research papers, so we're not seeing much of a gain on a task like this. BioASQ is another example of a very difficult assessment, but it's but it requires reading abstracts of papers from the last 10 years. And so it turns out many of the language models can do that because they've been trained on abstracts, but not full text papers over the last 10 years. Great. Okay. So in my last few minutes, I just want to mention, I said that we can make the input to language models, we can make the input to machine learning models be language. I showed you we did it with the material synthesis, we did it with the compound names. You can also do it by having LLMs write code, which is then input. But what about getting language out of these machine learning models, right? So that's one of the big questions, is that when you have a machine learning model make a prediction, it just makes a prediction. There's no explanation attached to it. And so a lot of my work, which I'm not going to talk about at all today, has been on how do we do explainable AI. And that is, how can we get a model that's predicting something to give an explanation? So we've written an article. I'm just going to share like one, way that we've done it is we've created these things called natural language explanations. And this is kind of built on the principle of local interpretable model explanations line. And that means that we basically take a prediction from a black box model and fit a locally correct natural language explanation to that. So we can say something like, our model tells us that the solubility of this molecule is minus 5.69, but then using our method, we can get out this paragraph of text, like the presence of a carboxylic acid and a hetero and non-basic aromatic group in this molecule, both positively correlated with its aqueous solubility. The carboxylic acid group is especially important as it's highly polar and form hydrogen bonds with water, blah, 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 blah. This text is generated by an LLM to go with the prediction. And so now what we can do is we can now have models which take language in, then we have models which can make language come out, and then we can put them together with agents. So I really think that we're on the cusp of a revolution in science, not just chemistry, not just biology, but in all fields. And the reason why is we're reaching a little bit of an S-curve, right? We've been building tools and we've been writing papers and we've been gathering data for a long time now. And that data has only really been able to be actioned by a few people, right? Any sort of PhD student, even if they're an amazing PhD student, they maybe use 3 or 4 tools for their one domain, and it takes a long time to figure out what those tools are. We're reaching a point now where we can start to compose all of the tools that we have available, all the data sets, all the research papers, and make them instantly accessible. And so that's why basically I've gone on a sabbatical to a nonprofit called Future House. Future House is a research institute in San Francisco focused on AI and biology. And our goal is to basically build this AI scientist, or this automated method of doing science, where we make basically the tools accessible to a large trained language model that knows how to use these tools to accomplish open-ended tasks and scientific discovery. So that's what I'm working on right now. Happy to talk more after this. So with that, I'd be happy to take any questions about any of this. And thank you to the funding sources and the students who contributed to this work. Thank you.
All right, thank you. Very interesting stuff there. I'm wondering what you think about data integrity in this case, because you mentioned like we've already have the issue of which are the good papers to read. And one thing people have brought up is that current ChatGPT and stuff make it very easy to make a whole bunch of very bad papers very quickly. And so there's the risk of the literature being swamped with a bunch of stuff that is possibly AI hallucinations. Do you have any ideas for how to discriminate what actually deserves to be used as input versus what is basically just noise and chaff?
Yeah, I get asked this question a lot. You know, I don't have a good answer right now. I agree with you that like if scientific literature is poisoned and we base a lot of insight on scientific literature, it won't work well. I have a feeling that we won't be the only people in trouble if scientific literature becomes poisoned. People will have to start working on that as well. One thing that we have thought about, we haven't really gone far, but what we have been working on is identifying areas where scientific literature disagrees. So one of the things this tool can do is you can give it like a state. I've showed it just in kind of question mode, but you can give it a statement and you can say, basically, identify what sources support or contrast with this statement and revise the statement to fit, the consensus. And that allows you to basically put in like sort of, a quantum of a fact, or just like one tiny statement from a paper. And then you can start to find out where's their disagreement or not. And that could lead to basically identifying flaws in literature, or it could lead to basically new science, because a lot of times, unexplained phenomena or disagreements in literature can also be a good track to follow for hypotheses generation. But you're right, like one of the underlying hypotheses is that the tools that we use, the data that we get from papers, the data that comes from open source data sets, and the papers that we get from journals, we assume all of that is, in good faith. But I think it's been a good model in the past. Maybe things will change, and if so, we will try to adapt.
I was just wondering, in these models like ChatGPT, and I'm no expert in this, but my concern looking from outside at this is that now we have things like ChatGPT. It generates a bunch of text, which is mixed with human-generated text, and the next generation of these models will use for training all of that, including the machine-generated text, and it's going to lead to exacerbating and deepening the biases and propagation of errors. Do people who develop this system think about it, and are there any safeguards in place against this happening?
Yeah. So this is something that's been discussed. I think there was a paper or two that built like a toy model to show that if you keep training on a model's output, it will eventually become maybe pathological. It's interesting because the AlphaFold paper itself was trained on its model output. It's actually one of the interesting innovations in the training in the AlphaFold paper is that they would actually train on their own output. So I think it's kind of a mixed, whether or not it's analytically correct, or it's always going to be the case that training on your output is bad. But you're right, like one of the most important kind of technical challenges right now with these very large language models is deduplication of training data and also filtering of training data. So when they train these large language models, right, one of the most important parts is making sure you get good quality data. And so these pipelines will deduplicate. They will also check to make sure data is natural language. And they also do checks on like perplexity or like the entropy of the data. So if you have a bunch of repetitive data, or you have like the statement, as an AI language model, I cannot answer, right? Like those sort of statements will naturally kind of filter out because they're low perplexity, or it's data that's not containing a lot of information. But even then, there's been interesting papers. Like there was a paper that showed if you train on musical notes or even DNA, you get an advantage when you go do language tasks with natural language. So even training on sort of bad data or repetitive data, as long as it follows sort of a grammar or has some kind of properties of language, it may not just be catastrophic. But you're right, it's something that we have to think about and consider is how do we make sure that we have good quality data keeping pace?
Yeah. So I had one question on the aspect of like collecting all of this data. So scientific literature usually summarizes only successes, but 95% of the effort that goes on in most labs is failure, at least in my group. So we don't document those in the papers in terms of like struggles that we have had to actually get to that point. But that is actually a very important piece of the puzzle, right? If you want to have models that answer these sorts of questions, what's your take on documenting those?
Yeah, that's a good question. This is part of that theme of like negative data or tacit knowledge that's, just known by people in the field. One of the remarkable things about these large language models is they have a very good prior. So like if you ask it about an experiment and there's maybe some missing detail, like the temperature that you culture cells at, mammalian cells, and you ask what the temperature is, it will usually get it correct, right? So a lot of times it does have a very good prior knowledge about these details. But you're right, like there are, especially at the cutting edge of science, there are papers that don't give you all the details, or you have to fill in some missing things for yourself. And I don't have a good answer for that. You're right, that's something we'll have to solve. I mean, there's this belief that there is to use this metaphor, there's dark knowledge or dark data, or dark science that we can't access yet. I think right now we are getting a lot of traction out of just accessing papers. Like we're getting pretty far. And then I think the next step is to access all of these data sets, put together by like NIH or the European Bioinformatics Institute, and start making those operable. And then you're right, at a certain point, we're going to hit a limit where we need to be able to capture knowledge that's missing. And it's interesting too, people thought that negative data would be required to build, very good machine learning models. But there's been a lot of, work, especially in LLMs, showing you don't need negative data. Like a lot of times the absence of data is enough for LLMs to recognize that that's like, an absurd example, right? Like if you ask, what's wrong with this sentence, and it maybe never saw that sentence, but it can tell something's wrong with it. Or you ask it about an experiment, it'll often give you, good advice, or like bad code, Although there's a lot of bad code out there, it's very good at identifying bad code because it's seen a lot of good code. So I don't know, I think it's an open question, but you're right. That is something we have to start thinking about as we get close to the human knowledge. A lot of things are not reported in the papers or are tacit.
Really cool talk. You had asked a question previously about like the explainability and like the motivations of why you chose certain methods. I was just going to ask, like, is there any explainability methods that you are really hopeful for, or is there anything that like really piqued your eye in the last few years?
Yeah, I think the method that we have been most excited about is something called a counterfactual. And it's kind of a different domain. So counterfactual is the smallest change possible to an input to change the output. Now, it's arguable whether that's an explanation or not. But what's nice about a counterfactual is that a lot of times people want to know why, because they want to change the outcome of something. Now, I think the talk we saw before, it was about explaining peptides. And that's just sort of a biophysical phenomena that we all are interested in, is like what part of the protein is responsible. But in things like if I want to understand why like my mortgage application was denied because of an AI algorithm, a counterfactual which says, oh, if you had made this income level, it would have been accepted. That's usually a better explanation or more actionable explanation for people than having, say, a report like, oh, it was 20% because of your income, 10% because of your, marital status or something, right? Then, of course, explainable AI is, it's like, what's your favorite cuisine, right? Depends on, what you're trying to accomplish, what your mood is, your personality is. The problem with XAI is it's tied up in humans, right? And humans are not, unfortunately, like, all the same, or fortunately, I guess. And so some people have strong opinions about what is an explanation. Some people have strong opinions about what is not an explanation.
Really cool talk. I'm going to ask a kind of different question. As a junior scientist, what do you foresee for the future of the career for a lot of students here in the future, where these large language models, these agents are start taking over? Do you see like a future where like PhD researchers can still prospect with these agents? Now, basically a PI can run a single lab with robotics with no need for human input.
Yeah, so this is an interesting question. I mean, there's one classical quote, which is like, robots won't replace chemists, or AI won't replace chemists, but chemists with AI will replace chemists. So basically, the students who embrace these tools, the junior scientists who start to use them quicker, will be more successful, and over time, it will be endogenous, I think is the word in biology. Now, I should say that, like, you see this already, or maybe you don't see this already, but in my group, when I have new PhD students, I make sure they all have GitHub Copilot and they all have ChatGPT. And basically, now I don't have to spend hours training students on how to make a Git commit or how to write a Python script. Students who go write research papers as native language or non-native English speakers, they can now take a paragraph of text and say, please give me critiques on if this sounds like a native English speaker, or what could I do to improve this writing? And the suggestions that come back are usually quite reasonable, right? So you can see this acceleration of PhD students that use these tools. And so I don't think it'll be like a role replacement. Now, with that said, there is some interesting dynamics. Like our tool, this paper QA, which I shared with you, it costs about 40 cents to ask a question. Okay, that's just the cost of the searches, the cost of the inference, the cost of the compute is about 40 cents. And the cost of a Google search is free, right? So you could imagine that if you have a setting where, every student can ask like 100 questions in lab A, and in lab B, the students can ask 1,000 questions or one question, right? You start to see this this unusual scaling where what used to become flattening, right? The internet was a big flattening thing. Now everyone can access papers easily. Well, maybe not everyone can access papers easily. Like everyone can, use email and communicate. Everyone can basically, use Google Docs or Google Sheets, right? Things got very flat. But I think we're going to see a reverse now where these AI tools become expensive and they're going to be concentrated in resource-rich laboratories in ways that just weren't feasible before. And so now, every lab gets PhD students, and, the PhD student at Harvard versus the PhD student, at any other university, the gap isn't that huge. But a PhD student with $10,000 of AI tools accessible to them, maybe that gap starts getting larger. So there may be some growing inequity. But I think right now it's just more about there are opportunities to accelerate training and accelerate science with the tools that are out there today with judicious use and, being smart about what's possible.
Right. I think there are no more questions. Let's thank Andrew for a really fascinating talk. And I think sort of nearing the end. And I think